Hello, my name is Dr. Giulio Antonelli. I work in Rome, Italy. And first of all, I want to thank Endoscopy, the editors, the reviewers, and all the team for handling and accepting our work in Endoscopy. Today, I will be commenting on our work, which is called The Clinical Implications of Computer-Aided Real-Time Size Estimation of Colorectal Polyps during Colonoscopy. I have done this work together with my team in Rome and colleagues from Milan and Bolzano, which I all thank for their outstanding support and help. So why did we do this work? Well, we know that sizing in colonoscopy, sizing of colorectal polyps is actually very important to determine patient care, because from size we determine the correct follow-up interval, we determine the possibility of using cost-saving strategies like leave in situ or resect and discard, we determine the best way of resecting those polyps. So a correct sizing and a correct inter-observer agreement is crucial for patient care. But we also know that between us, endoscopist physicians, we are quite not precise in determining polyp size, and often we tend to disagree. And we have also seen in recent years a huge increase in artificial intelligence-based system that can aid endoscopists during different moments of our procedure. We have seen AI for detection, we have seen AI for characterization. Today, we see AI for size estimation. And it's important to say that in this work, we aimed to evaluate the performance of the first ever fully image-based AI system for size estimation. And this is a significant difference from the previous AI size estimation systems, which always relied on a physical tool to actually help the estimate of the size. So this is the crucial difference, and I think is one of the most important points that set us aside from previous studies. So we enrolled 225 patients and 465 polyps total, and our AI tool re-evaluated all the full endoscopy videos and estimated the size of each and every polyp that was included. Overall, the accuracy was very high, with nearly 86% of concordance with expert endoscopist, which is the main benchmark that the ESGE sets for this kind of AI systems. We have also seen that when looking at diminutive polyps less than 5 millimeters, which comprise, of course, the majority of the data set of the training of the AI, but also the majority of the polyps we find in colonoscopy, the accuracy went up over 90% and went over 93%. Very importantly, we also looked at the clinical implications of sizing, and we found that using the AI-based system correctly assigned the right follow-up intervals and the right cost-saving strategies in over 90% of cases. So, in summary, we can say that a new AI tool for image-based size estimation of colorectal polyps is safe and effective and compares to expert endoscopists. And so it can be seen as a further step towards standardization of colonoscopy. There is some more work to be done. We have to increase the accuracy of the system for polyps more than 10 millimeters, so large polyps, and we have to validate these results in real-time clinical trials, possibly randomized and possibly used in conjunction with the other AI-based tools to closely mimic clinical practice. I would like again to thank all my colleagues and all the team at Endoscopy, and thank you very much for accepting our work and letting us expand its diffusion. Thank you so much.